Hi everyone, today we are going to talk about overproduction of proteins in bacterial system. One is the most crucial part in the life sciences medicine, particularly for molecular biology research. Why is overproduction is so important? Because many proteins of academic interest, some are medically important. Meanwhile, we can acquire valuable and functional proteins by that technology. So, what is overproduction? Overproduction is a technological process of generating a specific protein, which is typically achieved by the manipulation of gene expression in an organism, so that it can express large amounts of a recombinant gene. There are three key steps to produce proteins. Firstly, we need to clone gene into an expression vector and then transform that into a suitable host organism. Here is bacterial system, which will be deeply illustrated in the following video. Finally, purified proteins we need. To build expression vectors successfully, we need to use two kinds of enzymes, restriction enzymes and DNA ligase. Nowadays, more than 200 restriction enzymes have been found, which mainly exist in the microorganism. Here, we mainly focus on one restriction enzyme, ECO1. As one of the most commonly used restriction enzymes, ECO1 can only identify a specific nucleotide sequence, GAATTC, and cut DNA molecules at a specific site between G and A. Then it comes DNA ligase to connect the two cohesive ends of complementary parts to make it a complete DNA molecule. Both of them make function on diester phosphate. After knowing some information, let's start with the principle behind overproduction of proteins in bacterial systems.
Now we are going to be discussing about protein overproduction in bacterial system. As you can imagine, there are often many times we will need a large amount of protein, but it can be difficult to get it from the original source. We often want to produce our human protein in bacteria. However, bacteria don't have introns, they just have exons. In order to take a version of the gene that's from humans and get it to be overproduced in bacteria, we first have to make something called cDNA. So we are going to take our cell of interest. The first step in creating the cDNA is we are going to isolate the mRNA of interest from the cell. And once we have our mRNA, we are going to add reverse transcriptase. So it's going to take the RNA and it's going to put together this double-stranded DNA that we are going to refer to as cDNA. The next step, of course, is to gather cDNA into the bacteria. We do this using plasmid. The plasmid is a circular piece of DNA that the bacteria can then take up and read. So we are going to cut up the cDNA and cut up the plasmid. And we are going to have oversins that are complementary. Then we add DNA ligase. It will create a phosphodiester bond between the cDNA and the plasmid pictures. And finally, we will get the cDNA insert into our plasmid. The next step, of course, is getting the plasmid into the bacteria. In order for the protein to be expressed, one common way is called heat shock. What happens is that the bacteria is heated up and then cooled rapidly, and this creates lots of little holes in the membrane, which allows the plasmid to get into the cell. Now the bacteria will naturally express this protein, so you can grow up the bacteria in large quantities and get a lot of the protein of interest. Now let's move on to the operation.
So here comes to the laboratory operation. In general, the listed 5 steps are required to contribute to the oil production in bacterial cell. At first, the mRNA, which can be translated into the protein of interest, should be found and be purified. Once the mRNA is fractionated, it should be reversed to cDNA, the essential material for PCR amplification. If the mRNA is hard to purify, it is available to synthesize artificial mRNA or cDNA according to the consequence of amino acids in proteins. Then we need to design suitable primers to start PCR amplification. Next, the amplified DNA should be cloned into the plasmids of bacteria. Before cloning, we should cut the plasmids with restriction endonucleases. It will be better if it is a sticky end because it is more specific for the DNA ligase to pair the bases. After these steps, modified plasmids are ready to transfect to the bacteria. There are two widely used methods to transfect the bacterial cell. One is known as macroinjection. This means to insert the modified plasmids into cell directly with the help of light microscopy. The other requires the cells be treated with a solution which contains calcium cation. Then the competent cells are ready to absorb the modified plasmids. The last step is to determine whether the protein of interest can be expressed successfully. Generally, there are genes to resist antibiotics in the plasmid to determine whether the plasmids exist in the bacterial cell. Of course, the antibiotics also exist in the medium to cultivate bacteria. Next, let's have a look at the application.
Hello, my name is Dmitri, and I'll be talking about an example of overproduction in bacteria systems, and that is the production of spider silk. Spider silk is spun by all spiders, and it is a protein polymer. It is extremely effective at trapping prey, and what it makes it so good at doing that is its mechanical property. It has exceptional strength, toughness, and elasticity. This has been noticed for its benefits, and research has been done. It was found that it is tougher than steel and Kevlar. Since it isn't a synthetic material, it is biodegradable, but it is also long-lasting. Okay. Unfortunately, spider silk doesn't have the same strength properties as Kevlar to stop bullets, so it probably won't be much use in military services. Uh, it does have many other uses, though. For example, it can be used in tissue engineering, in and repair, since it is material found in nature. It can also be used for drug delivery as well as cosmetic products such as skincare. And finally, it has been used as nanofibers in industrial materials. However, unlike spider silk from silk or silk from silkworms, spiders cannot be farmed due to their territorial nature, and if attempted to be farmed, it leads to them attacking and eating each other. To make a beautiful outfit like this, it takes about 8 years to gather the material, with around 1.2 million webs needed, enough time to get 2 bachelor science degrees. Research into which species produce spider silk most effectively through recombinant DNA was done on E. coli, uh, Pichia posteris yeast species, silk larvae, tobacco, soybean, potato, Arabidopsis plants, and on hamsters, mice, and goats. This research shows that E. coli was the most effective at producing the spider silk with 300 milligrams per liter per cell cycle. However, it was found that recombinant spider silk had weaker mechanical properties than native silk, so we will have to wait and see if the product can be improved in quality.
As we know about the application of spider silk, I'd like to introduce the bacterial protein expression system, E. coli. Bacteria are rapidly and simple system of expressing recombinant protein because of the short doubling time. The media and required to culture them are not expensive, and the method adapted to scale up bioproduction are straightforward. E. coli is the most widely used host system since there is ample knowledge about its genetics, genome sequence, and physiology. The genetic manipulation is easy, and it also grows to high densities and is suited for large-scale fermentations. But one thing we need to be careful with is that the cell wall of E. coli contains toxic proteins, and the expression protein may have to be extensively tested before use. Then here is a picture of the procedure of how to express protein using E. coli expression system. First, use of compartment E. coli cells to take up DNA sequence of interest. Second, integrate the DNA into bacterial genome or circularization of the DNA sequence to exist as a plasmid. Then select transformed E. coli using a selection marker like antibiotic, and then expanded selected E. coli to a higher scale in a protease culture media. In the end, isolate and purify intracellular or secreted proteins. About the features of E. coli expression system, low-cost culture method, easy purification, good stability, flexible system, can carry plasmids with multiple promoters, tags, and restriction sites, easy to scale up and produce higher yield of protein, wide application.